All right, the next macromolecule that we're going to study is lipids. Lipids are interesting. They kind of vary in structure. They're going to be ultimately large, nonpolar molecules. Because they are nonpolar, will this make them soluble in water or insoluble in water? So before you move on, will they dissolve readily in water or not as nonpolar molecules? The answer is they are water insoluble molecules. They do not like water. They are insoluble. They are hydrophobic. Now, the interesting thing is that due to their very large hydrocarbon chains, their hydrogens are covalently bonded to carbon, are nonpolar. They tend to not form hydrogen bonds with water molecules. Because they do not associate with water, they do not readily break down in water. If you don't associate with water, how much harder is it going to be to break down the molecule using a hydrolysis reaction? You need water to break that molecule apart. So, because these are water insoluble, they do not like water, they're going to be used for our long-term energy storage. They're not going to readily break down. They're going to be able to hold on to that energy for a long time. Now, the interesting thing is lipids actually have a lot of functions, way beyond just long-term energy storage. They're also used in structural components. They are used to protect vital organs. They're used in heat retention and cell-to-cell communication. They're used for protection of various substances. They're used for insulating our nervous system. There's so many functions for lipids. They're very, very important. They make a vital component of the cellular membrane. So many purposes. But we're going to break them down and study them in 3 categories. They also form cell membranes. And we can subdivide them into 3 categories: triglycerides, phospholipids, and steroids. Triglycerides are going to serve as our primary energy storage molecules. They're going to be used primarily for energy storage purposes. And they come in a couple of different varieties. Fats are going to be one form. They're going to be the triglycerides found in animals. They're going to be used for long-term energy storage and insulation. They're often going to be found just below the surface of the skin. This helps retain body heat as well as being used for energy storage. Oils, on the other hand, are going to be the triglycerides found in plants. These are going to serve the same purpose. They're going to be used for energy storage, but they come in the oil form. Think of like vegetable oil, canola oil. I'm blanking on some other oils right now. Corn oil, sunflower oil. All of these oils are plant-based, and they're going to be found in that more liquid form. Phospholipids are an essential component of the cell membrane, and their unique structure makes them ideally suited for semi-permeability. Now, we didn't mention it, but there are also waxes, steroids and waxes. Waxes are going to also serve a protective function. A lot of times, waxes can be antibacterial or antifungal. They also serve in water retention. So not only does that waxy coating on the outside of your apple or the waxy coating on the outside of your grape, not only does it keep it fresh and plump and keep all the water in, it also prevents bugs and organisms from getting through. Another fun fact, the cerumen in your ears, in other words, earwax, has some insecticide components to it, or it's will actually deter. What's the word for deterring bugs? Not necessarily insecticide, doesn't kill the bugs, but it does prevent them from coming near. I am drawing a blank. Well, all right, brownie points to the first student who can email me with the term for bug deterrent. It's like bug spray. What's bug spray? It's a deterrent. Maybe there isn't a word. I don't know. I'm drawing a blank. But anyway, your ears are a warm, dark place. There's no air coming in or out. They're actually beautiful home for bugs, but we don't have that many bugs crawling into our ears because they are deterred by the presence of earwax. So there you go. Fun facts. We're going to take a little bit closer look at triglycerides. I've kind of already mentioned lipids don't really form polymers. They don't really form long chains. They're still macromolecules. They're still very large molecules. This right here, very large molecule, but it's not necessarily a chain of many repeating units. We actually have repeating chains of the same unit. What we actually have is two different units. We have a glycerol molecule and then three fatty acid tails. So we're going to look at the structure a little bit. We have three fatty acids bonded to a single glycerol. What type of reaction would you use to bind glycerol to the fatty acids? Are you going to use a dehydration synthesis or a hydrolysis reaction? What are you going to use if we want to assemble this molecule? Triglycerides are specifically made up of three fatty acids in one glycerol molecule. Each fatty acid contains a very long hydrocarbon chain. When I say hydrocarbon chain, I simply mean that it is made up of carbons and hydrogens. These long hydrocarbon chains are going to be nonpolar. They have no partial charges. They have no affinity for water. Each one of these is going to end with a carboxyl group at one end. This COOH over here at the end. This is where we are going to bond to our glycerol molecules. Now the interesting thing is, here we go. Examples are fats and oils. Both fats and oils are considered to be triglycerides, but they can be classified as either saturated or unsaturated fatty acids. And here's the interesting thing. The difference between a saturated and unsaturated fatty acid has to do with the presence of double bonds. If there are no double bonds, then we say it is a saturated fatty acid. By saturated, we mean that it is holding as many hydrogens as it possibly can. So you have one carbon bound to another carbon and holding on to two hydrogens. This carbon's bound to another carbon and holding two hydrogens. Each one of these carbons can hold on to two hydrogens. However, when you throw a double bond in there, suddenly there's only room to be attached to one hydrogen. So there's fewer hydrogens attached to this chain. It is unsaturated. Now the interesting thing is that the that saturated fatty acids tend to be solid at room temperature. Because they have these long straight chains, they can pack in very, very tightly at relatively warm temperatures. These are going to be things like fats. So butter, and they're typically going to be of animal origin. Fatty acids like butter or lard or are going to be solid at room temperature, whereas oils coming from plants are going to be liquid at room temperature. Coconut oil is actually an interesting exception. Coconut oil is of plant origin, but it is actually a saturated fatty acid, which allows it to be solid at room temperature. All right. Diets high in animal fats have been associated with circulatory disorders. The concern is that these saturated fatty acids might accumulate in the bloodstream. So saturated fatty acids have no double bonds between those carbons. They're solid at room temp, tend to be from animal origin. Unsaturated fatty acids have a double bond. This double bond actually creates a kink in the chain. And look at how much space there is when there's no double bonds. These are nice and closely packed. Once you throw a double bond in there, we suddenly create a kink in the chain. This prevents close packing, and it prevents our molecules from becoming solid at room temperature. So they tend to be liquid at room temperature and tend to be of liquid at room temperature and tend to be of plant origin. Nearly all animals use fat as their long-term energy storage. These fats tend to store a lot more energy than carbohydrates because of all of these carbon to hydrogen bonds. Just a quick reference. If you've ever heard of the term hydrogenated oils, this is where we take an unsaturated fatty acid, like an oil, plant oil, and we add hydrogen atoms, or we add hydrogens and we break the number of those double bonds that are going to be found there. So for example, shortening or margarine, vegetable shortening, those are of animal origin. They used to be liquid animal oils, but they've been hydrogenated. We've added hydrogens to them. We've removed those double bonds and allows them to be solid at room temperature. It also will increase their shelf life. Trans fats are fats that were once upon a time unsaturated and have become saturated through that hydrogenation process. So another very important lipid that I want to look at before we stop today is going to be the phospholipids. Now, phospholipids are extremely important. They are very similar in structure to triglycerides. They do have a glycerol group and they do have fatty acid tails. However, they also have a phosphate group that's been attached to that glycerol. So we have one glycerol molecule. It's going to make the molecule. Now, the interesting thing is, by adding this phosphate group, we actually can create a polar molecule. This glycerol head up here is going to be polar. What does polar mean? Does anybody remember? I'll give you a hint. It has to do with this little negative charge over here in the corner. Once we have a polar molecule, what else do we know about it? We know that it is hydrophilic, and if we remember, what does hydrophilic mean? Hydro, meaning water, philia, to love, means that the hydrophilic region really likes water, readily associates with water, loves water. Here's the interesting thing. We do have this polar head, but we also have these nonpolar tails. These nonpolar tails do not like water. They are going to be hydrophobic. Hydro, water, phobos, to fear. They're actually going to point inward, and this is what's going to help hold the membrane together. We're going to spend a lot of time looking at the plasma membrane, but we just want to touch on it for a moment. So here's actually what the plasma membrane looks like on the when it surrounds your cells. So down here we have animal cells. This looks like a tissue of probably looks like some skin tissue where we've got all these animal cells smashed up right next to each other. If you were to take a small cross section of this membrane right here and look at it close, you would actually see that that membrane is made up of two layers. On the outside we have our water-loving hydrophilic heads, and on the inside we have our water-fearing hydrophobic tails. The extracellular fluid, the fluid on the outside of your cells, is going to be mostly water. The intracellular fluid, the cytoplasm of your cells, is also going to be mostly water. Is mostly water. So what we're going to find is that the water-loving portions, the hydrophilic heads, are going to point towards the outside of the cell and towards the inside of the cell. And what we find is these hydrophobic tails are going to be making up the middle region. One other thing that I want to point out about this nonpolar hydrophobic tail is that it does contain one saturated fatty acid chain and one unsaturated fatty acid chain. This allows for close packing, so that way we don't have too much gap or too much space between our phospholipids, but it also allows for fluidity. Because we don't have these fatty acids, our cellular membranes aren't necessarily solid at room temperature. They're also not necessarily liquid at room temperature. They're sort of halfway in between, and it allows that plasma membrane to be fluid, move freely. So this is very important to cellular function. We're going to come back to this when we study the structure of cells, but I just wanted to spend a little bit of time looking at the structure of the phospholipid. And the last thing we're going to talk about in the category of lipids is going to be steroids. Steroids are going to be composed of 4 fused rings. They do vary in structure. They're very different from triglycerides or phospholipids. But they still are considered lipids, and they have very important purposes. They're going to vary based on their functional groups, and this is something that's pretty interesting. The functional group is going to be a small attachment that's going to completely alter the purpose of that molecule. So, for example, estrogen and testosterone are examples of steroids that are found in males and females. However, both of these arise from the precursor cholesterol. Now, you may have heard of cholesterol. Cholesterol has some very negative connotations. It can be a precursor to a lot of health issues if you have too much cholesterol building up in your bloodstream. However, cholesterol is very important. It is of the utmost essential component of the plasma membrane, but it also is a precursor to other steroids. So it's vital to the cell membrane. It helps regulate the flexibility of the cell membrane, but it can also be used to make other sex hormones such as estrogen and testosterone. All right, and I would like to point out or have us take a quick look at cholesterol. Here's cholesterol here. However, all we have to do is just make a few slight modifications and we can make the hormones both estrogen and testosterone. They're very similar